Welcome to the first in our two lectures about the tools for examining brain structure and function. This one will focus specifically on the tools used. The next lecture will focus on Roger Sperry, one of the more famous researchers who helped develop the use of some of these tools. Today's lecture was written by me, Chris Gall, and is based upon the College Board course outline for AP Psychology version 1. As always, you should feel free to stop this lecture if needed to take notes or to go back and rewatch all or part of it in order to clarify your understanding. So the learning target provided by the College Board for today's lecture is that scholars can recount historic and contemporary research strategies and technologies that support research. Specifically, we'll be examining a combination of research techniques like case studies and split brain research, and data collection like imaging technologies and autopsies. And really in English, what this means is just that you can explain a little bit about what these are and how they work to help support research in the brain. The first one we're going to talk about are case studies. And to quickly review what case studies are, they're the in-depth study of individuals in the hope of deriving principles that can be generalized to the larger population. The damage caused to the specific case studies are typically accidentally done, or at least they were not intended to cause the results that they did. Some of the more famous case studies when we're talking about brain research include Phineas Gage and HM. Phineas Gage, you'll remember, was a railway worker who accidentally drove a spike through his frontal lobe. And as a result, we learned the role of the frontal lobe in helping us to control our emotions and having a filter on what we say. Okay. HM is a completely different case. His is the one where the gentleman with epilepsy had a lobotomy to try and cure it, and as a result, lost the ability to make long-term memories. HM is a particularly fascinating case because he was able and willing to be studied for something like 50 years. So he generated a lot of data across a number of different ideas. Case studies are one of the oldest ways to study the brain and its functioning. Okay. The next one is split brain research. And split brain research is a particularly fascinating field. In this case, the brain damage is intentional and people know that they're selecting to have damage done. Okay. Typically, this occurs when they've got something like epilepsy. And epilepsy occurs when a storm of electricity happens in the brain, causing the rapid firing of nerves and a temporary inability to participate in the world. This can look like what we traditionally think of as seizure, which is to say a grand mal seizure, where someone is lying on the floor and their whole body is shaking for some length of time and they may or may not be foaming at the mouth. Or it can look like what are called petit mal seizures. Petit mal seizures, as the name implies, are smaller and harder to notice. They can often look like someone is not paying attention or engaging in repetitive motions. Split brain surgery happens when grand mal seizures are occurring regularly and cannot be controlled medically, specifically with those patients who are at risk of more serious harm because of the seizures. In a split brain surgery, the corpus callosum is cut, preventing the two halves of the brain from communicating with each other. When this happens, each half of the brain processes information independently of each other, unlike your brain and mine, where the two halves are constantly communicating. Scientists are then able to design studies to help better gauge what each half of the brain can do that is unique from the other. These differences are not nearly as distinct as popular culture would have you believe. There is no such thing as a left brain or a right brain person, for example. But they do exist. A spoken language is controlled by the right half of the brain, but language in general is processed by both halves. So these studies often rely on the fact that each half of the brain processes information from the opposite half of the body. Thus, scientists will design studies in which each half of the brain processes slightly different information and then ask them to show or tell what they processed. The results can then be used to illuminate better what each half of the brain actually handles. And I'll link a video that'll show you a little bit about some of the research around this, specifically some of Gazaniga's research around split brain surgeries and patients. So imaging techniques on a very literal level are taking pictures of the brain, okay? Only each one of these pictures shows different types of things. EEGs, for example, show the electrical activity in your brain and thus can be used to locate areas of brain damage by showing where nerves are not firing properly. MEGs work similarly, but instead use the magnetic fields that the electricity in your brain creates. CT scans are literally just x-rays of the brain, and much like the x-rays of your bones, can be used to show areas of severe damage, like lesions. PET scans track blood circulation and activity by showing how glucose is consumed in the brain. For a small organ, it's only 2% of your body mass. It consumes a lot of calories. Your brain consumes around 20% of what you actually consume in a given day. MRI scans expose your brain to a serious magnetic field and then disrupt the field in order to trace how the atoms react once they're no longer aligned. In so doing, it can take very detailed pictures of the softer tissues of the brain and show where things are perhaps not functioning the way that they should, right? Or where there's damage. fMRIs function similarly, but they track blood flow and thus brain function instead of brain anatomy. So lesioning is when there's significant brain damage that has happened. It's often done to try and solve to solve a specific problem by killing off small areas of the brain that are malfunctioning. So, for example, they might lesion your brain to try and control movement disorders like Parkinson's disease, which can cause really significant tremors in those who suffer from it, and they get worse and worse across time. This is a rarely used treatment just because of the potential for larger harm, right? Because it can be very easy to lesion the wrong areas. They don't like to do that very often, right? And the last one we're going to talk about are autopsies. And autopsies occur after death. Specifically, they're the dissection of a body in all or in part to help determine what happened to a person. For our purposes, it's specifically happening to the brain. Okay, so autopsies can be used to diagnose significant disorders and larger problems like Alzheimer's disease or chronic traumatic encephalopathy. And that's the one that's been in the news a lot because it's commonly found in boxers and football players that cannot otherwise be diagnosed. So autopsies can also be used to help see if people with certain traits have damage to certain areas of the brain. For instance, serial killers often get asked to donate their brains to science so that they can be dissected and studied in order to see if there are common types of brain damage that cause certain behaviors, like violence. So, you were successful with today's lecture if you can identify the methods and tools of brain research, including case studies, split brain research, imaging techniques, lesioning, and autopsies to help figure out how the brain works. As with all lectures, if you did not understand part of today's lecture, or if you were not clear on all the parts, please go back and rewatch all or part of the video. It's been a pleasure talking with you today, and if you have other questions, please bring them into class and I'd be delighted to answer them as best I can. Until then, be well.